Hello! In this series of videos, I will introduce what the Stein lab does and why we are interested in what we do. In this video, I will explain our research on axons and axonal signal integration and our paper on bioarchives entitled Control of Sensory Ectopic Spike Initiation by Descending Modulatory Projection Neurons. The authors are Carola Stedler and myself. I am Wolfgang Stein and I run the Crab Lab at Illinois State University. Many neurons possess a dendritic tree, a soma or cell body, and an axon. I'm going to draw a simplified version of a neuron here, with the dendrites connecting directly to the cell body. Dendrites and cell body receive inputs from many other neurons, often from several thousands, in a process that we call signal integration. At the axon initial segment, all signals are collected and generate a sequence of action potentials that travel unidirectionally along the axon. The axon can be quite long and can be myelinated or unmyelinated. I'm showing an unmyelinated axon here, meaning it is not wrapped by glial cells. On their way to their targets, many axons pass the paths of other axons, or they travel through areas of high synaptic connectivity that we call neuropil. Eventually, the axon reaches the terminal, the output region where it transmits all collected information to its postsynaptic partners. The axon is mostly seen as a conduit to transport information. However, there are many indications that the axon is much more than that and may interact with its environment or other neurons on its way to the terminals. Yet, we know very little about these interactions and how they affect the axon. However, such influences have the potential to bypass all previously processed information and to directly inject new information into the axon by masking, competing with, or adding to that information. Essentially, signal processing in the axon could short-circuit all inputs from the dendrites. Our lab studies so-called ectopic action potentials. These are action potentials that are generated away from the axon initial segment in the middle of the axon. We found that they are indeed generated in close vicinity to neuropil regions where many synaptic interactions happen. Ectopic action potentials are easily recognized because, in contrast to action potentials elicited at the axon initial segment, ectopic action potentials travel in two directions, towards the terminals and antidromically towards the soma. We know very little about how these ectopic action potentials are generated and what their function is. What we do know, though, is that ectopic action potentials have been implicated in several pathologies such as epilepsy and chronic pain. Our lab has evidence that these ectopic spikes are under neuronal control by neuromodulatory neurons, and that they indeed occur in areas of high synaptic interconnectivity. In other words, there seems to be a signal integration in the middle of the axon, which gives other neurons direct access to the main information carrier of the neuron, namely the axon. This is potentially a very efficient and direct way to add or modify the function of the receiving neuron. Our lab studies when neuronal signals affect axons and ectopic spiking, and where these influences stem from, which neurons control ectopic spiking and interact with axons, and under which conditions does this happen. Lastly, we would like to understand how ectopic spikes are generated or modulated. In other words, what is the mechanism by which action potentials can be generated in the middle of the axon in order to bypass all signal integration that happens in the rest of the neuron. Thank you for watching.